Now we're going to look at the results of the carbohydrate fermentation. So in the previous lab, you inoculated either the 3 or 4 tubes for the fermentation. You put 1 bacteria into each type of sugar. You did the 9 or the 12, okay? You had 4 bacteria or 3 bacteria that you put into 3 types of tubes, okay? Now we have some results here that I wanted to demonstrate. So this original tube right here is what they look like to begin with. They're red, there's no gas, maybe a darker red than that, okay? Now this tube right here and this tube right here are essentially the same color. The difference is this tube has bacteria growing in it, okay? It's cloudy. So the first thing that you want to look at, if it didn't change color, look for growth. Well, even if it did, always look for growth first. And you can see growth because it's cloudy, it has turbidity, there's some sediment, bacteria everywhere. Now this right here is positive for growth, but negative for fermentation because it did not change color. That means that this particular bacteria does not ferment sugar, it uses it for normal cellular respiration. It goes through oxidative processes where it goes through cellular respiration, not fermentation. So make sure if it didn't change color that you look for growth first. All right? Or, gosh, I'm not doing well today. Okay, in all tubes, look for growth first because you can't read a result until you've seen that it grew. Okay? All right. The next thing that you're going to look for is color change. So obviously these two turned yellow. With phenol red, yellow means acid. So when it broke down the sugar, instead of going through cellular respiration, it went through fermentation. And with fermentation, you get products like alcohols and acids. So both of these produced acid. Now I do see growth in the bottom. Both of them have sediment. Okay? I don't know if you can see or not, but this one, the Durham tube, has no gas in it. This one has a gas bubble right there. It's a couple of centimeters long. This one produces acid and no gas. This one produces acid and gas. All right, so those are how you read the results for the carbohydrate fermentation.